what we did learn by working on mycorrhizal fungi okay, so yeah, we have been lucky enough to get 2 genomes for 2 ectomycorrhizal symbionts over the last 5 years. So I will try to summarize what we learned by working on these genomes, and that's the work of 2 consortia, the Lacaria Genome Consortium and the Tuber Genome Consortium. So I will try to summarize in 25 minutes the work of 100 people. That's always very challenging. So what are these ectomycorrhizal symbionts? Let's go to the woodlands. I would like to bring you to the forest. And most people, when they walk in the forest, they see the big trees. In fact, we are mostly interested by mushrooms because a large part of the mushrooms that you are collecting during fall are in fact interacting with trees and forming mutualistic symbiosis. There are about 5,000 of mycorrhizal species interacting with trees in temperate forest ecosystems. 2/3 of them are basidiomycetes, 1/3 of them are skomycetes. As you see there's a wide diversity of mushrooms. Some of them are very good, like the black truffle of Perigord. Some are some nasty mushroom that you should avoid eating. But these mushrooms are in fact the fruiting bodies of an underground mycelium which is interacting with trees, especially root tips of that trees. And as you see there, the wide diversity of fungi are able to form a wide diversity of ectomycorrhizal symbiosis. This is a new organ formed on the root system by the interaction between the fungal mycelium and the root tips. Again, it's a mutualistic symbiosis, and the 2 partners are gaining from that interaction. But it's very crucial to understand that interaction because most of the fungi in soil are in fact symbiotic fungi. We did a 454 survey of the metagenome, or the species present in the forest soils, and as you see there, in only 1 gram of soil, you could find more than 1,000 species of fungi. And most of them, at least the most abundant of them here, the red arrows, are ectomycorrhizal fungi. So these fungi are there in the ground. They are very important for the forest ecosystem. They are very abundant. And we know for a long time that these fungi play a key role in the nutrient cycling in that forest ecosystems. They are also promoting the plant growth, for example, seedlings in forest nurseries, but also bioenergy trees in large plantation. They may have a role in bioremediation and detoxification of toxic compounds. How do they act to promote plant growth and play that crucial role in the nutrient cycling in forest ecosystems? As you see there, this small birch seedling is interacting with one of these ectomycorrhizal fungus, Bacillus involutus, and you see here that all the root tips are just interacting with the fungal mycelium, and the fungal mycelium is really enshielding the root tips. There is no contact anymore between the root cells and the soil. All the nutrients will move through the plant, the fungal cells to the plant cell. And from that root tips, there is an extensive web of mycelium growing underground and prospecting the soils, scavenging nutrients, nitrogen, phosphates, and other micronutrients. And all these elements are moved back to the root tips. In exchange of that duty and task and tough work, the small seedlings is providing, and the big trees are providing photoassimilates to the fungal mycelium. So it's really a mutualistic symbiosis where the 2 partners are gaining from the interaction. But as you see there, the fungal mycelium should be able to cope with the life in the material, organic material, the wood decay, the leaf decaying materials, and also able to interact with the root system. So we expect that these genomes, the genomes of these fungi, are very plastic and very complex. So again, the main role of these symbiotic fungi is to promote nutrient movement between the soil, here like nitrogen and P, moving through the fungal mantle through the mycelium living in between the plant cells and toward the root cells. And in exchange of that nitrogen and phosphate, carbohydrates are moving from the central cylinder to the parenchyma, then to the mycelium, and providing energy and carbon for the extramatricial mycelium. So what we have tried to do over the last almost 20 years is to fish out the signals, the gene and the protein involved in that interaction and controlling the development of the symbiosis between root tips like poplar root tips here and the mycelium. As you see there is a drastic impact of the mycelium on the root morphology. You have very big clusters of root tips, probably induced by the secretion of auxins by the mycelium. So the morphology of the plant is drastically modified by the mycelium, and we know very little about the signals which are exchanged between the two partners, and we know very little about the gene networks and protein networks involved and able to induce these drastic changes in the root system and in the mycelium. As you see there, the mycelium, when the mycelium is entering in contact with the root system, very quickly it is aggregating on the root surface, forming a mantle sheath, and then the mycelium is able to penetrate between the root cell, but never within the root cell, to form the hearting net, which is a place where the symbiotic exchanges are taking place. So again, we want to characterize and identify the genes which are involved in that interaction. So a good way to do that is to sequence genomes. So we have sequenced 3 genomes of different symbiotic fungi. Lacaria bicolor, which is a basidiomycetes, the black truffle of Perigord, or tuber melanosporum, which is an ascomycete, and we are still trying to sequence the genome of Glomus intraradices, which is belonging to the endomycorrhizae, fungi, a group of fungi which is mainly interacting with weeds and herbs and different agronomic plants. So what did we learn from the sequencing of the Lacaria genome? What are the basic features of the Lacaria genome? So first, the size of the genome is one of the largest fungal genome sequenced so far, about 60 megabases in 10 linkage groups, probably corresponding to 10 chromosomes. About 40% of the genome is coding for protein coding sequences. We found about 29,000 putative proteins. Many of them have orthologs in the other fungal genomes, but still there's about 1/3 of the Lacaria genes which are specific to Lacaria, and as lineage-specific genes, they may play a role in the interaction and in the symbiosis. A large part of the Lacaria genome is also formed by transposable elements. This was really one of the first surprises in analyzing this genome. The very large abundance of transposable elements. It is believed that in fungi, usually you have 1, 2, 3% of transposable elements. In Lacaria bicolor, we have many. Almost 1/3 of the genome is made of transposable elements, and as you see there, they are scattered all over the genome in red, and you have a lot of protein coding genes between these highlands of transposable elements. And we are willing to understand what is the impact of this transposable element on the plasticity and the evolution of the Lacaria genome. So a lot of transposable elements, a large set of genes, more than 29,000 genes, which is again very large, very abundant for fungal genomes. And we were also amazed to see that many duplications took place in that genome. We found more than 2,000 duplicates, different type of duplications in the genome of Lacaria, segmental duplication, tandem duplication, and probably the transposable elements are involved in that kind of duplication. And we also analyze, as we've seen earlier in Christina's work, we analyze the multigene family in the Lacaria bicolor by comparing the number of multigene family in the sequence basidiomycete genomes, Lacaria, Coprinopsis, Phanerochaete. So Coprinopsis is a saprotrophic fungus, Phanerochaete is a white rot fungus, Cryptococcus is animal pathogen, and Mucilego is a plant pathogen. And Lacaria is only the single symbiont analyzed so far. As you see on that tree, we have many multigene family in Lacaria, and most of them are in expansion compared to the other fungal genomes. In red, you have the expanding gene families, in blue, the contracting gene families, and in black, the ones which are not changing. So Lacaria is really a part of the rest of the sequence genomes by having a large set of expanding gene families. So a lot of duplication and expanding gene families. So we expect that this expansion of gene families have a role in the adaptation to symbiosis through that gene duplication and expansion of gene families. And we assume that within that expanding gene family, and specifically in the lineage-specific gene family, we may have the genes playing a key role in the symbiosis formation. So we are testing that hypothesis currently. So the best way to confirm that these genes belonging to expanding family may play a role in the symbiosis formation is to do gene expression. So we design an image gene array having the 29,000 protein coding genes plus a few hundred of transposable elements, and then we compare the expression of these gene repertoires in the different types of cells, tissues, the free-living mycelium, the mycelium interacting with poplar roots, and the mycelium interacting with Douglas fir roots, so the symbiosis. And as you see there, nicely, the symbiosis is inducing drastic alteration in the gene expression. More than 10% of the genes in the fungus is up or down regulated by the mycorrhizae formation. So when we screen the list of the up-regulated and down-regulated genes, many of these genes are belonging to secreted proteins. And I don't know if you can see clearly on that one, but on that slide, we assume that this up-regulated secreted protein could play a role in the interaction between the two partners. You should see there the fungal cell with the cell wall, and there the root cells where the fungal hyphae is landing. And between them, the two partners, we know that there are a lot of small secreted proteins which may be involved either in the attachment of the fungal hyphae on the root surface or maybe in the control of the plant gene expression. So when we list and when we look at the up and down regulation of the small secreted proteins, we get that kind of heat map where in red many genes are upregulated in the mycorrhizae. In green, a lot of genes are downregulated in the mycorrhizae. But you can see there that within the upregulated genes, we have a lot of genes which are also induced during the fruiting body formation. So the symbiosis formation and the fruiting body formation are inducing the same kind of upregulations regarding the small secreted protein. But still, we have there a cluster of small secreted proteins which are only involved in the mycorrhizae formation. So we have been focusing on that small secreted protein, the SSPs, and specifically on the one which is the most highly upregulated during the mycorrhizae formation, this small protein called MISP, for mycorrhizae-induced small secreted protein of 7 kilodalton, which is strongly upregulated during the mycorrhizae formation. You can compare here the level of expression in the Douglas fir mycorrhizae and the poplar mycorrhizae versus the free-living mycelium. So in the free-living mycelium, that MISP7 is almost barely detectable. In contrast, in the mycorrhizae, the signal corresponding to this transcript is almost saturating the array. So it's a highly expressed genes in the symbiosis, and it's highly upregulated. So we decided to investigate the putative role of this MISP7 SSP. This is MISP7 sequence. You have there the secretion signal. And we found in this MISP7 protein sequence two motifs which are related to the RXLR cell entry motif, which are found in many Phytophthora and may be involved in the uptake of the protein by the host cell. So when we knock out by using RNAi the gene corresponding to MISP7, we just decrease the mycorrhizae formation. You have there the wild type, the number of mycorrhizae, the percentage of mycorrhizae during the interaction between poplar and Lacaria bicolor in the wild type. And when MISP7 is knocked down by RNAi, you have almost no mycorrhizae formation or barely detectable mycorrhizae formation. So clearly, the inactivation of MISP7 is precluding the formation of mycorrhizae, meaning that MISP7 has a key role in the interaction between the two partners, in the control and the formation of the mycorrhizae. So what kind of biochemical role could MISP7 have? First, we immunolocalize MISP7, so we raised antibodies in rabbits against MISP7, and we use these antibodies to immunolocalize MISP7 in the different tissue during the mycorrhizae formation. Here you have no expression and no accumulation of MISP7 in the free-living mycelium. You have the control root there, and you see that as soon as the mycelium is interacting with the root surface, we can observe the accumulation of MISP7, and this will be confirmed till the end of the mycorrhizae development after 12 weeks of contact. So MISP7 is highly regulated, highly expressed, and also accumulating in the tissue in direct contact with the roots. And when we zoom in the cells which are at the interaction between the mycelium and the root cells, you can see there the accumulation of MISP7, and we have observed the accumulation of MISP7 in some of the nuclei of the root cells, suggesting that MISP7 may be entering the root cells. So to confirm that, we synthesized MISP7. We labeled the synthetic peptide using FAM, and we then incubated root seedlings in the presence of MISP7, and we observed the entry of MISP7 in the root cells, and as you see there, most of the MISP7 is targeted to the nucleus. To confirm the nuclear localization of MISP7, we incubated suspension cells in the presence of the synthetic peptide, the labeled peptide. You can barely see there that the nucleus is there, stained by DAPI. When we incubate in the presence of MISP7, there is a strong accumulation of the synthetic peptide in the nucleus, and this is a merge pictures between the DAPI staining and the incubation in the presence of MIS7. And as you see there, the entry and nucleus targeting of MIS7 is very rapid, and after only 20 minutes, most of the almost 80 to 90% of the nuclei in the suspension cells are labeled by MIS7. So MIS7, released and secreted by Lacaria, is entering the poplar cells, is targeted to the poplar nucleus. What are the motifs which may be responsible for that cell entry and nuclear targeting on MIS7? So we inactivated, I remind you that MIS7 has a signal peptide and two motifs which are close to the RXLR cell entry motif. We knock down, we replace the RXLR motifs by using alanine, and so I don't know if you can see it, but when the two motifs are there, the MIS7 is entering the poplar nucleus. When we inactivate the 3' motif, there is no entry of MIS7 in the nucleus. In contrast, when we inactivate, when we replace the 5' motif, MIS7 is entering the nucleus. And when we knock down and replace the two motifs, MIS7 is not able anymore to enter the root cells and the poplar nucleus. So this means that these two motifs are probably involved in the cell entry and the nuclear targeting. So this is the current model on how the interaction is working between Lacaria and poplar is that one, where we assume that a flurry, a series of small secreted proteins are released by the fungal cells. Some may be involved in the attachment between the two partners, but as we've seen for MIS7, some of the SSPs are able to enter the poplar cell or the host cells, and there are probably able to trigger changes in gene expression. So this kind of interaction between SSPs, effector-like SSPs, and plant cell has been shown in many times for plant pathogens. But this is a first demonstration that small secreted proteins released by a symbiotic fungus is able to enter the plant cell and able to modify and change the expression of that host cell. So to summarize what we obtain on the Lacaria genome, which may explain what are the features which may explain the mycorrhizal formation, we found that the large set of transposable elements may play a role in the genome plasticity of that fungus. We've seen that the symbiosis could also be explained partly by the expansion of multigene families. That lineage-specific multigene families has probably a role in the interaction. We found that effector-like SSPs are really crucial for the interaction and the mycorrhizal development. I didn't mention that, but we also analyzed the carbohydrate active enzymes, the enzymes involved in the polysaccharide degradation in Lacaria, and surprisingly, Lacaria has a very low set of cellulases and all the carbohydrate active enzymes, probably to avoid plant defense reaction. And we also observed an increased expression of transporters. So these features are clearly important for the mycorrhizal formation, and we would like to confirm that by sequencing an additional genomes. So we have sequenced in collaboration with the Genoscope the black truffle genome, and I will cut a long story short just to summarize the data we have obtained on the black truffle genome and in the comparison with the Lacaria bicolor genome. Regarding the genome features of the black truffle, we found again a very large genome. This is the largest genome for fungus, for fungi, 120 megabase. We have an even larger set of transposable elements compared to Lacaria. More than 50% of the genome is made of transposable elements, but we have a very low set of protein coding genes. So a very huge genome, but with a very compact gene repertoire and protein coding genes. And a large set of them have orthologs in the other fungi, but still we have about 20% of the genes which are lineage specific. In contrast to Lacaria, we have no expansion of gene families in the black truffle. In contrast of Lacaria, we have even a contraction of gene families. Compared to the other ascomycetes, sequence ascomycetes, there you can see that only 269 gene families are expanding, whereas more than 5,000 of gene families are lost and lacking in the tuber genome. So a very different picture than we have obtained on Lacaria. When we looked at the transcriptome during the mycorrhizae formation, here we were not able to find any small secreted proteins. Lacaria tuber is not using SSPs to interact with the plant cells. In contrast, we found cellulases, lipase, lacase, peroxidase, enzymes probably involved in the degradation of the cell wall during the penetration. So again, a transcriptomic pattern very different than the one we have obtained with Lacaria. So to summarize what we've seen in the black truffle, as Lacaria, we have a large set of transposable elements which may play a role in the plasticity and, of the genome. But we were not able to find expansion of multigene family. We were not able to find many lineage-specific multigene families. There is no SSPs, no effector-like SSPs in the black truffle of Perigord. But as in Lacaria, we found very few cellulases or plant cell wall degrading enzymes, and we found an increased expression of transporter. So clearly, by comparing the two genomes, we have obtained two different stories. So this would mean that the ectomycorrhizal symbiosis is a very ancient innovation which developed probably several times during the course of the mycota evolution using very different toolkits. The two fungi are interacting with the plant by using very different gene networks and mechanisms. I would like to thank my collaborator working on the MISP7, and I would like to thank also the different organisms funding that research, and specifically the JGI and the Genoscope who provided us with the two genome sequences. Thank you very much.
Okay, any short questions?
Yes. Did you find MIS7 homologs in saprotrophic fungi, basidiomycete fungi like Coprinopsis?
No, this is really a Lacaria-specific transcript protein. There is no ortholog in any of the sequence basidiomycetes.
Okay, well, one final question.
Just curious, did you look to see if you could pull out some interaction partners for your small screen protein?
You mean if we did immunopooling to identify the targets?
Yes.
We did that, but in fact, we just got the sequences last week, and most of them are ribosomal protein or glycolysis enzymes, so probably the immunopooling fails. So we have to repeat the experiment. But we are working on it.